Hi, welcome to a Physionic detailed study analysis. Today we're going to be discussing a lot on autophagy, specifically going into a study that tries to show if autophagy occurs in the brain primarily, but we also discuss hepatic or liver autophagy. So I'm going to show you a series of different graphs and data as well as some images of what autophagy really looks like, because people kind of throw the word around kind of aimlessly. And then they show maybe some diagrams of some pictures of people having drawn it out. But I'm going to actually show you under a microscope what autophagy looks like. So it's going to be a really cool episode as we go through this detailed study analysis. If you're not familiar with who I am, my name is Nicholas Verhoeven. I'm a PhD candidate in molecular medicine. The lab that I work in for my cell biology research to finish my PhD is actually in, we do at least half of our work is in autophagy, and I've actually been published as a researcher in autophagy. Specifically the journal Autophagy, which is called Autophagy. How many times can I say autophagy? And the paper that we're going to be looking at in this study analysis comes from that particular journal, Autophagy. So those are my credentials. That's what we're going to be doing. Let's go ahead and jump into it. All right, so as always, I will walk you through step by step, but the topics that we're actually going to be covering are: does autophagy occur in the brain? Does fasting induce brain autophagy, which I know is a hot topic for a lot of people? And what does autophagy actually look like? As I mentioned, I'm not just going to be showing you diagrams, pictures, but actually show you microscope images of what autophagy looks like. And then for my insiders, if you're interested in ad-free experience of this detailed breakdown as well as the full detailed breakdown, also looking at fasting on liver autophagy, then hop on over to my Physionic Insiders page where you'll have exclusive access to this as well as all my other detailed analyses as well as seminars and all kinds of other good stuff, which is hopefully going to help you. Well, it should help you in your health decisions, and it also comes with a community, which is really fantastic. So if you're interested, then hop on over to that right now, and I'll have links for it, so you don't have to get a truncated version. But if you're just interested in these three questions, then let's move on forward. All right, so as I mentioned, this is going to be coming from a study. This study is called Short-Term Fasting Induces Profound Neuronal Autophagy, and it comes from, as I mentioned, the Journal of Autophagy. That's definitely a word I'm going to be saying a lot. So, as always, anytime we're going to be doing a detailed study analysis, a deep dive into studies, then we have to actually understand how they went about this study. Now, granted, we can't study brain autophagy in humans, at least not based off the technology that we have at the moment, because, well, we'd have to take brain slices. We have to take the brain out or take sections of the brain to actually do a lot of these experiments. So we have to do them in animals and use them as a proxy for how we learn about how our autophagy works, because we use mammalian models. So in this situation, we're talking about mice. So this is a super simple design so far, but there's a genetic component that I'll get into in the next slide. So they have mice that are normally fed, which is really simply they get to eat whenever they want. They get to eat their normal food. And then they had mice that were fasted for 24 hours, and then they had mice that were fasted for 48 hours. Now, granted, is 48 hours of fasting for mice the same as 48 hours for humans? No, it is not. So trying to make that parallel is inaccurate. However, just look at it from the perspective of just fasting as a whole. And we're going to be able to determine if fasting has an impact just generally on autophagy, specifically in the brain, and what that looks like, as I mentioned. So really simple design. Just had these three conditions for the mice, and then they would kill the mice and then remove sections of their brain and of course their liver, if once we get to that data for the insiders, and then from there end up doing a bunch of measurements on those brain slices, and we're going to be able to see all that. But there's another added component to this in that the mice, some of the mice, most of the mice, were homozygous mice, meaning that they had a genetic mutation, or I shouldn't say mutation, a genetic anomaly that was added to them, a genetic change that was added to them that led to a change in the DNA of these mice. So what was that change? The change was to express or to add a gene that expresses or leads to the realization, the production of a protein known as LC3. And I'm going to show you what LC3 does in the next slide. But for the time being, just know that LC3 is related to autophagy. But for more concise and better measurements of LC3, it's nice when you can tag that protein with another protein that is known as a green fluorescent protein, and that's what I've got here displayed. So these mice express, they produce an LC3 protein with a tag that is green fluorescing. So then if you look at it under a microscope, as we will see, you will see a green fluorescent tag that's attached to LC3, and therefore then we can tell when LC3 is present. And LC3 is then a strong proxy of autophagy. So these mice are in every other way normal, and that's why they had a condition where they didn't do this genetic manipulation to be able to tell the difference between the two. And so these mice that have this genetic anomaly are perfectly normal in every other way, except that they express, instead of just LC3 without this tag, they express it with a tag. And that's going to help us see some of the data that we look at. So a brief background, and good gracious, this is incredibly brief, background on autophagy. So what normally forms is you have what's known as a phagophore that forms, which is kind of a vesicle inside of our cells. And this ends up maturing and elongating, growing, to the point where we have the incorporation of this protein, this LC3 protein. There's also many other proteins that get incorporated. They're known as ATG proteins, or autophagy-related proteins. And these all play a factor, but the main one that we're focused on is LC3. So LC3 will be typically converted from a particular form of LC3 known as LC3-1, and it will be converted to LC3-2. So LC3-2 is then the one that's actually incorporated into the autophagosome, or the phagophore that ends up turning into the autophagosome. And this autophagosome, which is autophagy, will then collect, with the help of LC3, different organelles like mitochondria, for example, will attach or grab onto misfolded proteins, protein aggregates, different lipids that aren't in their correct orientation, whatever it might be. All kinds of things. Autophagy has many different functions, but we'll just generally consider it cellular cleanup. It essentially goes through, it's like a huge, think of it like this. Think of it like have you ever seen a whale when they consume a ton of krill, which is their food source? They just open up their huge mouths, and then they essentially suck up. I mean, not that I'm a whale expert or anything, but what looks like they suck up all this krill as they go through the krill. And that's essentially what autophagy is. It's these big phagophores that open up and just gobble up all this stuff, and then they close into a vesicle, into a closed circular or semicircular vesicle, which then gets turned into what's known as the autolysosome. So the autolysosome is another vesicle, will come in, and it has components within it that are able to destroy all the parts of the autophagosome. So the different mitochondria, organelles, destructive like proteins that aren't working well, old proteins, whatever it might be, that need to be destroyed and have been captured in that phagophore, in that autophagosome, will then blend or bind together, as you can see here, with what's known as the lysosome, and that combination of the two leads to the production of the autolysosome. And the autolysosome here then contributes all these destructive proteins, these destructive enzymes, and changes the environment within that vesicle. Now, when it does that, that leads to the mass destruction of all these different components. So all these different structures that were found in there immediately get destroyed. So they get broken up, they get cut up, they get any sort of damage that you can imagine occurs to them, and then they get released back out into the cell, and there's a bunch of different fates that occur to them. They can be recycled, they can be kicked out, they can be used for other functions. There's a number of different fates that can occur to them. So that's a basic understanding of how autophagy works. What is autophagy? But that is not actually offering the answer to the question that I mentioned, that I'm going to show you what autophagy looks like. I will actually show you what it looks like, not just these diagrams. But let's go ahead and go into this data a little bit. All right, so here what they've done is we've got these brain slices, and they've taken out, they're looking specifically at neurons. So they're looking at particular brain cells. Let me remove my image just for a second so that you can see what we're looking at here. Remember, this is the LC3. So the blue is the LC3 with the green fluorescent protein that's tagged. And I'm showing this as an image just so you can see this is the real image, and this is my representation of what we're talking about here. All right, let me bring myself back now that you've seen that. Okay, so they've flattened these neurons, they've flattened these brain cells, and then they've added a stain. So the stain, what it does is it stains specifically for nuclei, or the nucleus of the cell, and I've stained it here as blue. So this blue stain enters the nucleus and gives us a distinction of, oh, this is a cell. So because not all cells, but in this situation with neurons, there's one nucleus. So each cell has one nucleus. So if you can literally just count them, right? There's one here, one here, one here, one here, one here, et cetera, et cetera. So you can tell how many cells are in a particular image. And then from there, they're also able to quantify the amount of LC3. Remember, it is a green fluorescent protein that's attached to it. What do we see? Well, we see when we combine, so here they've got it in black and white because everything that's illuminated here is LC3, so it's just black and white. However, when we combine the two images, so this is the exact same image, but with the addition of the nuclei, we actually see it in green. So you can just focus on these panels right here. And then they've got a quantification here that we'll get to in just a second. But for the time being, we'll just focus on these three panels. So we've got our normal fed, and we can see that there's certainly some autophagy occurring. And this is something that I've said many times before, although you don't hear it very often in the mainstream, not because it's against the grain. Like, this is actually true that in any situation, you have autophagy occurring. People ask this question of how do I activate autophagy? And I have a video on that. But the truth is autophagy is always happening, no matter what. If you're consuming food, there's autophagy happening. Doesn't matter what you're consuming, there's autophagy happening. Doesn't matter what age you are, doesn't matter what sex you are, none of that matters. Autophagy is occurring if you're alive. So there's some green that's, of course, going to be present because autophagy is occurring. Now, what's interesting is these mice, after they've been fasted for 24 hours, you can see, just contrast these two images of the amount of green that's present. And of course, they end up quantifying it over here. But the amount of green that's present is rather substantially higher after 24 hours of fasting compared to no fasting. Now, what's interesting, what's really intriguing to me is then when they look at the 48 hours of fasting, you would see, you let me know what your thoughts are, but for me, when I look at this, I think, it seems like there's a decrease in the overall green that's happening here. And what I think is happening is actually there's a much greater concentration in specific segments of the cell. So let's look at this cell right here, right? So we've got our nucleus, and you can see these branches coming off of it. So those could be dendrites, they could be axons, which are different components of the cell. So right, this is the blue, and then you've got the dendrites right here, and you've got the axon coming all the way down here, and the axon terminals. So we're looking at that right here. So you can see that there's a bunch of what are known as puncta, or these like segments that are filled with autophagy markers, or LC3 specifically. So I think that instead of having general LC3 just kind of all over the cell, they're concentrating in particular segments. Now, that's just my interpretation, because they actually end up quantifying this through mathematical and computational means. So it's not like they're just looking at the image and saying, oh yeah, there's more autophagy, because yeah, I mean, I would argue that there's less autophagy in this situation. But when they actually quantify it, which is over here, or some of the quantifications are over here, I haven't added all of them because they get a little bit redundant. But overall, we see, let's say, the number of autophagosomes. Remember, that's this, the mature version of this. The number of autophagosomes is increased after 24 hours. So that's if we compare no fasting versus fasting for 24 hours, and then we fast for 48 hours, we see that there's this increase again in autophagosome number. So there are more autophagosomes per cell with the more fasting that occurs. Now, the area of the autophagosomes along with the perimeter also increase. So the size of the autophagosomes, I'm going to show you an actual image, a zoomed in image of what that looks like a little bit later on. But you get this increase in the size, not only the number, but also the size of the autophagosomes. So the takeaway here is that with fasting, we do see increases in autophagosome formation in neurons, specifically in brain cells. Now, what I found really, really cool, and I wanted to share this, and I shared this on my Physionic community page, that they did a 3D render of what this would look like. So here, the blue is the nucleus. So again, a stain for the nucleus, and this is just a 3D render. But it's, again, the single nucleus, and here we've got multiple because we've got multiple cells. And this general green, or this kind of light blue, is the different axons and dendrites that are coming off of the cell. But really what we're focused on is this kind of light green over here. So it's indicating these light green puncta, or concentrated versions of LC3. We're seeing this again here because the 3D render is showing that there's far more of these green segments than in the normal fed condition. There might be one right here, but for the most part, you see a lot more. So you see a greater concentration of these autophagosomes, specifically LC3, concentrating together. So this is a stark contrast between the two. And really, I mean, everything else about the cells looks identical. It's just that you get more autophagy that's occurring. So then they moved on to looking at what are known as Purkinje fibers. So Purkinje fibers, let me look at my notes real quick. So, Purkinje fibers are brain cells as well. They're also neurons, but they're a slightly different type of neuron. And here they're again staining for, so they don't have this labeled all that well, but they're still staining for blue is for the nucleus. They have orange, which you can't really see all that well either because it's so taken over by the green fluorescent protein of LC3, which sort of dominates everything. But you do see some yellow, some kind of orangish color here, and that's a pan neuron marker. What does that mean? It means that it's a marker that identifies specifically neurons. So they're just making sure that where the LC3 is occurring is in neurons, because that's what they're trying to measure. So they're taking brain slices. So this is the overall view. So if you were to zoom out and just look at the brain slices, this is what it would look like, right? And then they zoom in, and within, this box, they zoom into this box right here. And then if they zoom in even further and look at just individual cells, so 1, 2, 3, 4, then we can see even more zoomed in versions of these cells. Now, this is still not fulfilling my promise of actually seeing an autophagy, like actually seeing an autophagosome. We'll get to that. And here again, we've got our green fluorescent protein, which is expressed right here, so within the cells, and we can see that's all extensive, so it's all over the place. So these fibers, these cells, as you can see, they extend way out. They branch out a ton. So out of a single cell, the kind of the head, or what's known as the soma, the cell body, is here, and then it protrudes everywhere. You have this mass release of all these different sections of the cell that branch out like tentacles almost, as you can see here. So we see that illustrated in the real version. So LC3, which is a little bit easier to tell here because they're only showing LC3 here, and here they're showing merged with the pan marker, the LC3 marker, the nucleus marker, and I think they also added, if I remember correctly, they ended up adding what's known as an astrocyte marker. Yes, red. They added a red marker for astrocytes. So this is all done through what's known as immunolabeling, but I won't go into that because otherwise we're going to get too off track. So, but astrocytes, just as an added component, are other cells. You can think of them like support cells for neurons. They do other functions as well, but astrocytes are also brain cells. So all of that is jumbled together in this merged image, but here we're just looking at LC3. They've just made it black and white so that any sort of lighter sections are LC3, and if you compare these two, you can see that that is representative. So what does this tell us? Well, it tells us that in Purkinje fibers, which are a specific type of neuron, that we see LC3 is still present. So we see that autophagy is still occurring in the normal fed mice. But then we also see that even if you were to compare these images, you can see that there's more widespread autophagy that's occurring. So it's extending outwards a lot more, that you're getting this stronger signal, especially when you get to 48 hours. I mean, it's almost completely green because it's so overpowering. And you can tell here, the soma is filled with LC3. It's just exploding with LC3 comparing, for example, this one, it's almost saturated by comparison to this, which is the strongest one in the normal fed. And even in one of these, which is also normal fed, you see just low levels of LC3. So this just confirms some of the other data. Now, something else that they wanted to do was look at mTOR. The reason why they wanted to look at mTOR, if you're not familiar with mTOR, mTOR is a protein. It's a master protein, just like I always call AMPK as a master protein, because it has such wide-reaching effects. It affects all these different cellular signaling systems. So mTOR will activate or phosphorylate, indirectly will phosphorylate a protein known as S6, ribosome protein. So this S6 ribosome protein leads to greater protein synthesis. So it leads to greater muscle, or not muscle, well, technically yes, also in muscle, but in this situation, in all cells across the board, that it leads to greater protein synthesis and greater cell growth. So what do we see? So this is shown, so they're showing the proxy, so the phospho S6RP, which is the S6 ribosome protein. And remember, this is an activated version by mTOR. So this is in normal fed and in our 48-hour fast, and they're indicating this in red. So the blue is, again, I believe it's the nucleus still. They may have changed it to something else, but that's not as important as looking at the comparison between LC3 and the phospho S6RP. Okay, so In normal cells, we can still see that mTOR is very active. It's quite active when the mice are eating perfectly normally. And we see that when we superimpose LC3 plus this mTOR marker, S6 phospho S6RP, that we see kind of a combination of the two, right? It's kind of orangish, some green, a little bit of red. But when we fast for 48 hours, look at how much reduction in red that there is and how much of an increase there is in green. And if you then superimpose both of them together, merge them together, there's suddenly a huge increase in autophagy and a massive decrease in mTOR. And that makes sense because mTOR is antithetic to autophagy. It's not saying that it's bad. I'm not making any judgments on what mTOR is in relation to the health or performance of the cell or anything like that. It's just the point is that if you want to activate autophagy, you have to lower your growth signals. That's the big takeaway. Okay, now, this is my promise to you. I mentioned that I was going to show you what an autophagosome looks like, and this is what it looks like. And there's a takeaway here that beyond just looking at an autophagosome. So here we're looking at what's known as electron microscopy. So electron microscopy allows us to get real resolution on within a cell. So here we've got a single cell, right? And we're zooming into these different quadrants, these different sections of the cell. Think about how incredibly powerful a microscope has to be to not just isolate, not just look at cells, not just isolate a cell, but be able to look within a cell and then to be able to look at the molecules that are accumulating within that section of the cell. Crazy technology, and I'm really excited for the future to see where this technology goes even further. Anyway, so this arrow is pointing to an autophagosome, which is this circular section. You can see that there's components within it that have been trapped. Okay, so this is normal fed, and here we've got with food restriction. Now, why are they doing this comparison? Well, the comparison is actually based off of this line. So here we're talking about 500 nanometers, incredibly small. 500 nanometers is indicative of this line. But the same one, 500 nanometers, is also indicative of this line. It's much smaller. So that means that looking at the relationship between this thing, this autophagosome, and this line, clearly the autophagosome is much larger when there's food restriction, when there's this fasting for 48 hours compared to the normal fed, because the line is much bigger in comparison to the autophagosome. And we see more examples of this right here. More examples of these autophagosomes, obviously pointed out by the arrows, although they seem pretty obvious to me, with these components within them that have been trapped. And then if we do a quantification, we see that the number of autophagosomes also increases in these Purkinje fibers. So these Purkinje fibers see a huge increase in the number of autophagosomes as well as the size of the autophagosomes, which is really just confirming through a different method what we were looking at here, right? We saw that the number increased in the general neurons that we were looking at, as well as the perimeter and the area were also increasing. So what are the conclusions up to this point? Well, the conclusions are that fasting does increase autophagy in the brain, as well as reduces mTOR-related activity. Now remember, the caution here is trying to translate that to humans. I feel pretty comfortable in saying that fasting does increase autophagy in the brain, but is 48 hours fasting going to lead to the exact same amount of autophagy that occurs for the mice? Probably not. Because you have to actually study that. You have to actually look at those parameters. You have to actually look at human beings. But it's really difficult for us to get brain tissue on humans because who's going to volunteer for that? So there are special circumstances where we can do it, but it's going to be incredibly limited. So that's really the only conclusion that we can make, that fasting increases autophagy in the brain as well as reduces mTOR-related activity. And if this applies to humans is speculative on my part, but I think it's got strong evidence that probably this does occur. Okay, so now we're going to move on to autophagy in the liver. Now for If you're just watching the public access version of this, then you'll have to hop on over to the Physionic Insiders if you want access to the data on the autophagy in the liver, as well as all my other detailed analyses and all that stuff in their complete form. And with that, I hope that you learned something. I hope that I was able to offer some useful information, or at least interesting information. And I'll catch you in the next one. Have a good one. Bye.